So first, let me thank the organizer. It's my first stay in this meeting and it's great. Thank you for gathering such a good meeting. What I'm going to present you today is a study about, yeah, why not, about complementary killing activities of two species of phages infecting Pseudomonas aeruginosa that were used in the context of phage therapy. And this work just recently came out in AAC. So this work was initiated through the contact with a small phage therapy company who wanted to understand better phage host interaction for their phages that they were preparing in a GMP state and distributing for compassionate use in France and also for some trials, clinical trials. Very curiously for me, these 4 phages that they gave us belong to only 2 species, but they had altogether a good coverage of a panel of 43 strains of clinical isolates of Pseudomonas aeruginosa. So we went on to characterize this relationship, asking 3 questions. What is the best growth medium for the loose 24 phage? Because at the onset of this work, it was not yet known what was the receptor for this phage. And then we were also interested in looking how these phages would work on biofilms. So this is how most of us start our experiments. We just grow phage and bacteria together in rich medium. And what we found with this loose 24 phage is that it was pretty unable to promote growth inhibition. You see maybe a slight decrease in growth, but very less marked than for the second phage species, LS1, that was doing a good job at preventing growth. But when we put the 2 phages together, we could see still a stronger reduction in growth. So we were confident the loose 24 phage was able to grow in this medium, but not to a large effect. So we were annoyed because we could not select bacteria that were resisting to this phage. So we went on and studied which growth medium could improve infection. And what we found is that minimal medium was very good to grow the loose 24 phages, because now, as you see, they inhibit bacterial growth as well as the cocktail of the 2 phages. And what we observed also is that now the phage was able to plaque and to make turbid plaques on minimal medium. And the phage was also able to propagate in the sputum medium, which is mimicking the lung conditions, and in partial anaerobiosis. So this allowed us to go forward and select phage-resistant mutants. But before to go to this panel, I want to insist here, because I think it's starting to be present in the community, but really the phage host range is medium dependent. And if you think about the phage you are selecting for therapy, it could be that those that you select on rich medium are not as active in the real condition of life of your infection. So what was the loose 24 receptor? We isolated phage-resistant mutants, sequenced them completely, and observed that all of them had a PSL mutation. I will tell you just right now what is PSL, but to convince us that this was a receptor, we just observed first that all these mutants were adsorption mutants. And we also found that by complementing these mutants with a gene that was mutated, we restored adsorption and plaque formation. So we were convinced that we were touching at the receptor. So what is this PSL? It's a surface and exopolysaccharide. It's a neutral polymer which has a cell-free and a cell-associated form. It is the main component of the PaO1 biofilm, and its regulation is pretty complicated, but it relies mostly on the cyclic DGMP. So we were intrigued, how can an exopolysaccharide be a phage receptor? So we wanted to quantify the PSL production in our different growth conditions because we found this phage was not growing the same in all medium. So we followed, there's one lab in the US that is a specialist of the PSL analysis. So we followed all the protocols very well, extracted the PSL that was bound at the surface of the bacteria after washing them. We also recovered the supernatants to see whether we had PSL in there. And then we do immunoblot of these PSL extracts on nitrocellulose membrane as if it was proteins, in fact. And you use antibodies. Here we had the chance to get human monoclonal antibodies that were really specific for the PSL. So we first did some controls to see that the experiment was working well. We could see that there was a linear range of response over, on a 25-fold range, which was not bad. And more importantly, that our PSL mutants were not or nearly not responding to the antibody. So we were confident we were going to dose our PSL. And here is what we found in minimal medium. It was very nice. Depending on the growth stage of the culture, we compared overnight, optical density 0.2 and 0.5. And here you have two independent repeats of the same experiment. You see quite clearly, I think, even in the back, that there is barely any PSL in overnight culture, but as soon as you grow bacteria exponentially, you get the PSL attached on the surface of bacteria, and we couldn't see any PSL in the supernatants. So we saw, okay, that's really pretty good conditions for phage growth. We understand why this phage is able to multiply in this condition. And the contrast with the rich medium was impressive because in this case, what you see on the left is that the bound PSL is mostly present in overnight saturated culture, but as soon as you start growing bacteria exponentially, there's almost much less PSL at the surface. And what we saw in the supernatant is that was a massive amount of PSL in the free form of the supernatant. So this kind of result could quite well illuminate why we had such a problem growing the phage in rich medium. There is less PSL on the surface, and it's mostly synthesized in the stationary phase. Now, because PSL is the main constituent of the PaO1 biofilm, we wanted to see if by any chance these phages were good at killing biofilm. We were completely new in the field, but we learned. We had colleagues in the lab who did some biofilm analysis. So we did mature biofilm grown for 16 hours with a plasmid expressing the GFP in the 96 well plate in a static system. Then we add phages at MOI around 2, and we monitor biofilm degradation by confocal microscopy over 2 days. So that's different from the dosage that people do. It's much more simple to dose the sugar of the biofilm, but thanks to confocal microscopy, you can really follow the live cells, not just the matrix. So once we have all these images, there's a long image treatment to project the image on a 2D dimension, and then to quantify the volume of all the bacteria in the biofilm. I show you just one end image of this 48 hours treatment with phage for one experiment of each condition. We did this 9 times in total. So when there is no phage added, you see a quite thick biofilm. The green surface is what you saw on the top of the biofilm, and the black section shows you the depth of the biofilm, okay? And when you add the LS1 phage, you probably see that the depth of the biofilm is reduced, but not much. With the Luz24 phage in this experiment, it seemed a little bit more reduced, more attacked. And the combination of phage was always giving a result that was quite more significant and convincing. But there is a but, because here I'm just showing you the last time point, and it's a very late time point, 2 days after phage addition. And what we were really surprised to see is that the kinetic of degradation is very slow, meaning that for the first 20 hours after phage addition, we didn't see any degradation at all. When there is no phage in the biofilm, you see that after 20 hours, anyway, the biofilm starts to decay a little bit. And the whole question is whether when you add phage, the decay is more pronounced. By doing this experiment 3 more times, we had sufficient evidence to be convinced there was some slight activity of the phages individually, and maybe a little bit more when you put the 2 phages. This was especially clear for the endpoint, but the kinetic was not magic at all. There is a paper that was recently released that showed very convincingly for different bacterial species that when you put a phage in a biofilm, it generates LPS that are activating biofilm formation. And I wonder if this is not what's taking place here for these first 20 hours. There is maybe attack and regrowth, and so you never see a real decrease in the biofilm. And that's probably why we will probably all agree that phage attack on biofilm is not something so easy to achieve. So to sum up, what I've shown you, why did I enter the topic by saying the phages have complementary activities? First, for 2 reasons I already showed you, that the LS1 phages, they lyse more effectively in rich medium, and the Luz24 phages, they lyse more effectively in poor medium in all the conditions where we had slow growth. So you can imagine on a wound or in the lungs, if you have bacteria growing at different speeds, it's probably good to have both kinds of phages. And what we found also is in some conditions, not always, but in some conditions, the combination of the 2 phage has an additive or a synergistic effect in biofilms and in liquid medium. There's a last thing I don't have time to show you in details, but I wanted to touch it because it's changing a little bit our view. I'm sorry because it's very low. Those in the back won't see what it is. But what we found is that if you first apply the LS1 phage, you will select for LPS mutant because the receptor of LS1 is LPS. And some of these mutants, the WZY mutants, they are sensitized to the Luz24 phage, meaning now what you see on the left here is a lysis curve of the Luz24 phage in rich medium on this mutant. So whereas on the wild type strain, the Luz24 phage is barely able to infect, in the mutant, it's becoming effective. And what we found is that, in fact, the LPS that are made by this strain are having a shielding effect opposing the phage infection. And what is very nice also is that our plaques, which were usually turbid, became clear on this LPS mutant. So if I have just 1 more minute to wrap up all the results in a single model that I would like to propose you, is if we consider the surface of Pseudomonas with a double membrane layer, on top of it you have the LPS, which are shown with this pink and red sugars, and we have drawn very delicately the PSL polysaccharide somehow hidden below the LPS to represent this shielding effect of the LPS. And the phage is probably in the wild type strain somehow stuck at the surface without the ability to infect because of this shielding effect, plus the low amount of PSL on the surface. I try to represent this also in the framework of the level of cyclic DGMP. I barely talked about it to you, but another paper just came out at the same time as our paper, who had much more results on this GMP level story. So when the medium is rich like this, you have low levels of DGMP, cyclic DGMP, so the level of PSL is low. And now, if you look what we have found in minimal medium and what the colleagues of this study published in eLife found in the flagellar mutant, in both cases, the level of DGMP is high, and as a consequence, the PSL level now raise on the surface and counteract the shielding effect of the LPS, so that now the phage is able to infect. And what is happening in the LPS mutant in rich medium, we still have this we measured, but I don't have time to show you. We still have very low level of PSL on the surface, but there is no longer this LPS shielding effect. So this is probably why the phage is able to infect again once the LPS is mutated. With this, I want to thank you and thank also the people who did this work. So in our department of Michaelis, this work was initiated by Maud Bio in my team and followed then by these other people. We had a lot of help of the team B3D and Romain Briandet, who helped us with the biofilm analysis, and the Faxiam company. Clarisse Plantadi did also a lot of work on this presentation. And with this, I will take questions.